Recording in progress.
One minute, 1 minute.
second.
Good morning, students. Am I audible?
Yes, ma'am.
Zoom is limited to 100.
Yes,
Hello, am I audible?
Yes, ma'am.
Yes, Zoom is limited or not? Fatima is here. Jacqueline, please unmute and tell me. Jacqueline. Jacqueline, am I audible? Akash UV.
Yes, ma'am.
Zoom is limited.
Yes, ma'am. I think so because it is live streaming.
Okay. Your classes will start shortly. Wait for a while, okay? Within 10 minutes, your classes will start.
Good morning, students. Am I audible? Hello. Am I audible?
Yes, sir.
Okay. So good morning. And today our topic of discussion will be learning and memory. So you've already gone through the sensory cortex, the motor cortex, certain subcortical structures like the basal ganglia and all. So this is one of the higher functions of a central nervous system, that is learning and memory. And as you would know, this is something that gives us a great advantage over all the other species or over animals, etc. So what do we mean by this learning and memory? What do we talk about? So let me ask you something. Okay, Lakshmi, could you unmute yourself?
Yes, sir.
Yeah. So Lakshmi, tell me, what did you have for breakfast today?
Kani,
And what about dinner yesterday?
yesterday I had beef and noodles.
Okay, good. So yeah, you can mute yourself now. So what was my point in asking this question? See, the moment I asked her what was her breakfast, the response was very quick. That is something that she had just been through, maybe in a couple of hours back. And then the memory of yesterday evening is important because it was something that she liked very much. And so it stayed in her memory. So all these things we store in our memory based on various aspects. Some things that we need immediately. For example, if I'm picking up my phone right now to call one of you guys, I'll just look up the, if I only have you on WhatsApp, I'll just open WhatsApp, just read your number, commit it to my memory and dial it. And by the time I finish the phone call, I would have forgotten it. I wouldn't remember it unless it's something that some number or some person's number that I really need to be remembering again. On the other hand, memories from my childhood, for example, say something like, yeah, if you talk about cashew nuts, the smell of roasting cashew nuts, I hope you guys know what that is. So when we roast, you wouldn't get that smell in our normal white colored packed cashew nuts, but the smell of roasting cashew nuts, if I get that smell now, or if you can get that smell now by those hand-picked cashew nuts that you get now, hand processed with a bit of the skin on it there, you'll get a bit of that smell. If I get that smell, I am reminded of my native place and the time when we roasted these on a small wood stove in the backyard of my grandmother's house. Why is that memory something that is so stable in my mind after all these years? This is something like what, 30 years back, something that has happened 30 years back. I haven't been to that place in more than 10-15 years now. We don't own that place anymore. And still, this one memory, the very smell of roasted cashew nuts, takes me back to that place. I remember very vividly that wood stove. What was near that wood stove? There was a chicken coop. I remember my uncle standing beside me, helping me, teaching me how to roast it. Why is that memory so deeply entrenched? If I am going to ask one of you guys, what did you eat day before yesterday evening, unless it's something very special, that the time that you spend with your friends or else, or if it's just something like your regular canteen dinner, you might not remember what you ate that evening. But I remember something from so long away. So this is a very complex process of learning and memory that our brain is able to achieve. Why am I associating, why do I remember things from my childhood? Because it was a very pleasant memory. It was a memory of very good times, because we went to that place only when schools were closed and it was vacation. So that is something that I'm going to solidly remember, probably till the time I start losing my neurons. Maybe old age will cause me to make them forget, but usually that will be a solid memory. So why did I tell all these things? I asked one of you guys a question. I picked out memory of that person. I gave you associations of certain my childhood memories and everything. In all these things, you have been made alert. Maybe you have started thinking of something of your memories. This again is a part of the retrieval of a memory also. I have made you guys alert. Probably you were sleepy from the last class. I do not know what your last class was, or whether you had a last class or not. Now you are alert. Now you are listening. It's a multi-pronged process. Your reticular activating system is right now active and alert. You're thinking about these things. You're thinking about what memories you have from 18 or 20 years back now, why it's staying there so strong, all these processes. So it's a very complex process. As such, it's a higher mental function. It involves those various areas that you already studied in the sensory cortex, in the various areas of the cortex, in the various cortical association areas, in structures like the basal ganglia, cerebellum, etc. All these parts are complexly linked to form these memories. As such, if you think of a computer, you know that whatever you have typed, let's say I have this PowerPoint open here. I typed up this PowerPoint on my desktop, and when I saved it, I know that it is stored in the C drive in my hard disk in my computer. It is stored as bits and bytes. It's stored as ones and zeros. This is not how memory is stored in our body. Our memory might be stored in different areas, depending on the kind of memory, what all it's associated with. If, for example, the cashew nut story I told you about, it has various components to it. It has an auditory stimulus associated. So whenever I get that smell of cashew nuts, I'll remember that. Or if I see a wood stove, that memory can be provoked. Or a similar situation where there's a wood stove and a chicken coop nearby, or that uncle, when I see him, that can provoke that memory. So multiple associations are there, and our body, our brain can very efficiently recover memories like that. Okay, so moving on. So this is what all we'll be trying to learn today. How do we define learning and memory? How do we classify memory? The mechanism of each types of memory. What is consolidation of memory? What are the intercortical transfer of memory, and the applied aspects to this topic. Okay. So coming to definition of memory. So the first paragraph you see here, learning is an acquisition of information that gives humans the ability to alter their behavior based on the experience, and memory is the retention and storage of that information. So you'll see that learning includes acquiring information, and where is this acquired from? It is based on our experiences, our various sensory perceptions, emotional perceptions, all of them together. From our experiences, we collect information, and this information gives, why do we collect this information? It gives us the ability to alter our behavior. That is what learning is. So it is acquiring information from our experiences for the purpose of altering our behavior. And is it enough that we just acquire this information? No, this information has to be stored if we have to use it later on. And there is a role of memory, and memory is the retention and storage of the information, and later on the retrieval of the information also. So you can understand from this that learning and memory are two sides of the same coin. You cannot have learning without memory, and memory without learning. You need to be able to remember what you learned to have memory, and you need to be able to acquire more information if any memory has to be created at all. Okay, so if we split it up, learning is the acquirement of information or knowledge by experience that results in alteration of behavior, and memory is the retention of that learned information and experiences. So going on, we'll classify memory into different types. So why am I not classifying learning? These are very interlinked. As we go through this classification, you will see where all learning is popping up, where all memory has a role. So I remember that I told before that about how information is stored on your computer, on your hard drive. It's as ones and zeros we know. We know exactly where it's stored, in what format it is stored, because we are the ones who designed it. Okay, but memory, how is it stored? Is it written down? Is it kept in your brain cells? Can somebody tell me how memory is stored? You've already gone through conditioned reflex, so you would have some idea of what is happening. Anybody, can you tell me how memory is stored in our bodies, in our brains? കേട്ടില്ല, ഒന്നുകൂടെ പറയാം. Can someone tell me how memory is stored? Synapses. Okay, good. So memories are mostly stored in synapses. This is not something that we have entirely understood, but from situations like our conditioned reflex, we realize that it is through increasing synaptic strength, increasing synaptic transmission, maybe creating more synapses, creating more neural connections is the way in which memories are created. When a particular stream of thought or a particular stimulus is coming in, as our senses pick it up, take it to our brain, is it perceived, and the response is based on connections that have already been made with other functions due to past experiences. This is a simple basic form of memory that we have understood. It has various components to various facts to it, like how the synapses are strengthened or weakened, the chemical changes and all are there. But basically the idea is it is through synaptic the connections that memory is stored. By increasing synaptic connections, decreasing synaptic connections, strengthening synaptic connections, or weakening them. This is the one area of memory that we have understood. This doesn't define everything. It is not 100%, but this is what we have understood so far. So how can we classify memory? Memory can be classified on two bases. One is physiological classification based on how the information is stored and recalled. And second is depending on how permanent that storage is. So as you can see here, the first classification based on how the information is stored can be classified as implicit memory or non-declarative memory, or also known as reflexive memory. So from those terms, from reflexive memory, you can understand it is something of a reflex process. And you know from the reflexes that you have studied that reflex is something that is not conscious most of the time. Our reflex actions are usually actions that are, you do not need to plan and act, rather reflex responses are made. So the same way, implicit memory, that there is no necessary need of awareness of a person. These include mostly skills that are developed, reflexive actions that are you have developed. We'll go a bit more detailed into that later on. An explicit memory or declarative memory is one that requires awareness, where you are aware of your surrounding, you're perceiving things, events happening around you, the sensations around you through your auditory, visual, olfactory sensations, etc. All that you have perceived consciously is used for creating memories. That is the explicit memory. So implicit is more of reflexive, where you needn't be aware of the memory that is being created, and explicit is you need to be aware. One thing you will require to remember, like all physiological processes, these are all ways we have found to classify them. It doesn't mean that they are discrete or they are independent of each other. Many processes will include both the implicit and explicit memories. Short-term memory, later on in the next classification, you see short-term, intermediate, long-term. Long-term cannot happen without short-term being there. So they are all interlinked. They are not mutually exclusive. So physiological basis is explicit and implicit, and permanency of storage, short term, intermediate to long term, and long term. So short term memory is that which lasts for few seconds or minutes. That is all. After that, you have thrown it away. One example is the example I told earlier. I just look at a phone number on a card, and I memorize it. I type it into my phone and I dial it. By the end of the conversation, I probably am not going to remember that phone number anymore, especially if I do not need that anymore. Then intermediate long term is the intermediate step in the conversion of the short term to long term. So these are memories that could be retained for days, maybe. Then long term is memories that are stored for decades or even for a lifetime, like the childhood memories we have. Okay. So going into each of these now. So implicit memory or a non-declarative memory or a reflexive memory, it is acquired through various forms of reflexive learning. And you have already learned some of these forms, that is the classical condition and operant condition. So the four forms of implicit memory that we know of is non-associative learning, associative learning, skills and habits, and priming. So what do you mean by non-associative learning? Means that these are learnings from simple or single stimuli to a single perception. That is, a person is getting one stimulus and or say one noxious, a painful stimulus, and based on that, a memory is created. I know that next time when I see a sharp object, I don't touch it. We'll go a bit more detail into that in the next slides. So that non-associative learning has two sides to it, habituation and sensitization. We'll go into that deeper. Second is associative learning. There you have the conditioning reflexes that you already studied. There is classical conditioning, operant condition. So there we associate an unconditional stimulus with a conditioned stimulus. So unconditional stimulus is where you already, that is something you do not need conditioning. That is something that is inborn in your, inherent in you. And by associating a conditioned stimulus with it, you create the response that the unconditioned stimulus used to exert to the new stimulus also. The Pavlov's experiments, and also in operant conditioning, the experiment with the mouse that you would remember, Skinner's mouse experiments. Okay, that you already know. So I will not be going into associative learning today because you've already dealt with that, is something in detail and has been dealt separately for you. Then we have skills and habits, skills like cycling or driving, or habits that we develop. So initially they take some practice to develop. So initially there is some amount of declarative memory formation involved there. So I need to practice how to ride a bicycle, or how to drive a bike or car, or how to swim. I need to, or even to juggle balls, or skills like that. Initially we need a lot of declarative memory there. Perception is involved. But later on, you start doing that reflexively. People who drive regularly would know that gear changes are things that we do not think much of. In response to a speed at which we are going, in response to a situation, we automatically shift the gears. That is how we do. So that is what skills and habits involve. So once it's set, it has become a skill or a habit, then it is no more declarative, but rather an implicit memory. It happens automatically. Then there's a term called priming. Priming means the ability for us to remember certain things by, you have already been exposed to that. For example, these words. When I say the word associative, you already know what it means. So when you're going to be, let's say I'm going to present one slide here, and I just use the word associative where I write ASSO. Those who have already studied memory will know that it refers to associative learning. If I write ASSO and put a full stop and then write learning, you would automatically know that it's associative learning that I'm talking about. So something that you've learned by previous exposure, later you can recollect even by seeing parts of it. The common example given is that of words. You see first 2 or 3 letters of the word, in the context you are able to make out what the rest of the word is. That is priming, is what it says. Okay. So going a bit deeper into that, in non-associative learning, I said there is habituation and sensitization. So non-associative learnings are simple forms of learning. They usually involve a cellular mechanism of learning. So the experiments done in this was in a snail called Aplysia. the gill withdrawal reflex was done. Basically, what happens is that you would have seen these snails and shell creatures. If you touch them, they immediately withdraw into the shell. So in the aplysia in water, if they give a touch, a tactile stimulus to the creature, it immediately withdraws its gills into the shell. That is what is seen. That is a normal response. But if this touch is repeatedly applied, it is seen that after some time, the gill withdrawal does not happen. It becomes slower. Then finally, it does not withdraw the gill when you touch it at all. This is habituation. That is due to what happens is that when you give repeated stimuli, the response to the stimuli, the reflex responses decrease over time, and finally they stop responding. So what could be the cellular mechanism there? We have seen the reason is that it is due to gradual inactivation of calcium channels. You would remember that how does neuromuscular junction or the NMJ synaptic transmissions happen? One thing you would remember that the action potential reaches the end of the neuron. Some neurotransmitters are released. They land on the receptors on the receiving side. This causes depolarization there. It causes opening of calcium channels, or rather the opening of calcium channels is happening there. In the presynaptic ending, when the depolarization reaches there, it causes opening of calcium channels. Calcium enters the synaptic membrane. This causes some process by which the synaptic vesicles that contain the neurotransmitters adhere to the presynaptic ending and release the neurotransmitters into the synaptic cleft, which is then picked up by the postsynaptic neuron, causing depolarization there and actions there. So what happens here is gradually, with repeated stimulus that the animal perceives as non-noxious, non-threatening to it, it perceives these things as inert stimuli. That is no consequences. Then finally, what happens is there is gradually inactivation of calcium channels, which decreases the intracellular calcium, decrease the release of neurotransmitters from the presynaptic terminal, and finally the reflex action gradually stops. So if you keep on stimulating the animal with something that is non-noxious to it, you see that finally the reflex action it was giving initially, it is going to be stopped. This is habituation. That it becomes a habit kind of, from that word you can understand. The opposite of this is sensitization. So here what happening is a presynaptic facilitation. We were seeing inhibition in the habituation. Here facilitation happens. In the same experiment, if a painful stimulus is being repeatedly given to the aplysia or the snail, the reaction to that becomes rapid. When a non-noxious stimuli, just a touch was given, after a few times it decided not to withdraw its gills. The withdrawal reflex was slowed down and stopped. But here, if you give a painful stimulus, when repeated stimuli are given, the reflex withdrawal becomes even more faster. So this could be a transient response, or if it is reinforced again and again, you will find that it can develop short-term or long-term memory also. That is, along with giving the touch stimulus to the aplysia, if you're going to give a painful stimulus also at that time, again and again you are going to repeatedly pair these. Finally, when you touch the snail itself, it will develop a sudden withdrawal of the gills, reflexive withdrawal of the gill sound. This is sensitization. Okay, so these are the two forms of non-associative learning we know of: habituation and sensitization. They are the opposite each other. In habituation, repeated non-noxious stimuli result in the reflex getting muffled, lowered, and finally stopped. In sensitization, the reflex becomes sharper or faster due to presynaptic facilitation. So moving on from that, we go into associative learning, classical and operant conversion. You already know about this, so I will not be going into detail into that. Then priming. As I said, it's a facilitation of recognition of words or objects by prior exposure to them. This is a function of the neocortex. Example, the improved recall of a word when presented with the first few letters of it. I already explained to you that is what priming is. So it is a function of the neocortex. Okay. We'll be later on seeing what all areas of the brain are primarily involved with the various kinds of memories. Then we have what is known as procedural memory, which is the same as the skills and habits I told you already. So once acquired, they become unconscious and automatic. That is, during the process of acquiring a new skill or habit, there is a lot of declarative memory involved. Your perceptions are involved. Your reflexes are being trained. But once the training is completed, this becomes unconscious and automatic. That is why it is clubbed or grouped together with implicit or reflexive memory. And this type of memory is mainly processed in the striatum, which is part of the basal ganglia. You already have learned, I think you are going through classes in basal ganglia now. So striatum, as you know, is part of the basal ganglia, and that has a very important role, skills and habits. Also, cerebellum has some role to do in this. So you can see here the various forms of memory that are involved and the various areas that are involved here. On the right now, we have come to only the non-declarative. So the bottom half of the picture, you can see non-declarative or implicit memory. We have procedural memory, priming, associative and non-associative learning. And below that, you can see the main areas of the brain that are associated with these different forms of implicit memory. So procedural memory, as I told already, striatum is the main part, but also cerebellum and motor cortex are also needed, because why? It's a skill, it's a habit, it's something that you have to do. So the memories that are already there are used to plan movements and execute them through the cerebellum and the motor cortex. Then priming, as we told, it's mainly a function of the neocortex. Then associative learning, you see here that amygdala and cerebellum are involved. What is the role of amygdala here? Amygdala, as you know, is part of the limbic system. So amygdala is involved with the emotional part of it, the emotional component of it. As we know, when you study associative learning in the classical conditioning and all, emotional part of it has a role. Especially in the operant condition, you see there is a reward or punishment part involved in that. So it is not just reflexive reactions. It is not just non-noxious stimuli. There are noxious stimuli also involved there. So there is the involvement of both rewards and punishments there. So the emotional component is also involved. That is, the amygdala and cerebellum are also involved. And non-associative learning, as we said, it's a simple learning, and it's mainly affected through the reflex pathways that we have. Okay. Moving on, the upper part of it I will show you again when we are doing declarative memory. You can write it down later. I hope you have got the lower part completed. Now going on to explicit memory or declarative memory. So far we were talking about non-declarative memory or implicit memory or reflexive memory. That is something where your the memory being retrieved and used does not involve your conscious attention. But here, explicit memory or declarative memory needs your conscious attention, your perception of the things that are around you. And it includes two components. One is the semantic and the other is the episodic memory. Semantic memory means memory of facts, that is concepts, words, alphabets, numbers, things like that. So facts, those things that are solid, discrete facts that are involved in semantic memory. Episodic memory or autobiographic memory is memory of events that have happened around you. So it is not discrete facts, it is but events altogether, events that you're remembering. So you can understand that from just knowing their definitions, that these are not again discrete. There are lots of places where these will intermingle. Episodic memory will have a lot of semantic memory also associated with it. And our brain, the complexity of connections in our brain is enough to be able to store these in discrete areas, but at the same time retrieve them to form complex memories in our mind. Okay. So that is explicit memory or declarative memory. It requires your attention, your perception, and can include both semantic and episodic components. So how is encoding of the explicit memory done? We saw already in implicit that various discrete parts of the brain were involved. Either in non-associative learning, there was just the reflex pathways were enough. In associative learning, we need some more part. So how is this explicit memory? How is it encoded? We told that it involves our perceptions and sensory. So all the inputs that are coming in through our sensory modalities, they are processed in various areas depending on whether it's short term, intermediate, or long term. They are processed in various areas and solidified. So the prefrontal cortex and the hippocampus and the parahippocampal cortex are very important components in this. This is something that we learned primarily by certain studies on a person known as patient HM. So this is an interesting case. Interesting on one side, yet tragic on another side actually. This person, patient HM as he was known, he suffered a trauma in his childhood. Around 6 years old, he suffered a bicycle accident. From then on, this person started suffering temporal lobe epilepsy. That is seizures that was triggered in his temporal lobe. So initially, he was having tonic-clonic seizures, then he was having repeated seizures, came to the point where when he was past 16 years or 17 years, when he had to start working, he could no longer hold a proper occupation because he'll have sudden blackouts, he'll have sudden seizures. So he was at risk to himself working around any kind of machinery. I think he was a factory worker or something. So he was losing his job and his livelihood. So in that point of time, the doctors working on him decided that since the temporal lobe is the cause for these seizures, they were able to identify, they decided to do a novel procedure in him, wherein they removed large parts of the temporal cortex and some associated areas with that also. The patient's seizures were relatively well controlled. He could try to live a normal life. He was not having very frequent seizures. Some were there. They removed both sides, the temporal lobes that were causing his seizures. So his seizure activity came down very, very low, like once in a month or so. But he developed this big complication, which was this guy couldn't create any more long-term memories. They found that he could remember things from his childhood. He's already around 27 years. I think it was 1953 that the surgery was done. He was 27 years at that time. He could remember everything from the past. He could learn new skills also. You could teach him to bicycle, or a lot of new skills could be acquired also. He could be proficient in those also. But if you ask him, what did you eat yesterday evening? Like I asked Lakshmi, he would say, I do not know. That is what happened to patient HM. And through life, and from then on, he couldn't perform, he couldn't hold on to a job also naturally after that. But he became a study subject his lifelong. And at the end of his life, he was in a care home and all. I just said that he could function quite normally. He was a person who lived happily also without any of these memories. You would think that it will be a terrible life without memories, but seems he was a person with a positive outlook on life. He lived a happy life. Ultimately, he died in an old age home in around 2007 or 8 something. He died. So he had a normal long life also. Only problem is he couldn't create long-term memories. So over these years, starting from around half a century, this person was studied by scientists and doctors. His behavioral changes, various changes they studied in him, and they found out, and he was the first source of information for us, that the conversion from short-term memory to long-term memory is primarily a function of the hippocampus in the temporal lobe. There were other areas also that was removed from him, but by repeated different kinds of program stimuli and questions and questionnaires and all, they were able to find out that the hippocampus is the primary region for encoding of memory, that is storing of memory from the short term. We told about like very short memories, like when we are trying to remember a phone number or something, and then after some time we forget. From that point to it becoming something that is committed to a memory that we'll remember later is mainly a function of the hippocampus, and it was mainly our main source of this information was from this patient HM. Later on, posthumously, after he's died, his name was released as Henry Gustav Molaison. His name was given. Okay. So here you can see that the declarative memory can be divided into semantic or factual memory and episodic or events memory. And the semantic memory, the main areas involved are the lateral and anterior temporal cortex and the prefrontal cortex. Whereas episodic memory, the main areas involved are the hippocampus, medial temporal lobe, and the neocortex. The memories of all these events of a particular event, for example, like I told you the example of cashew nuts. So all those parts of it, the visual part of it, the olfactory part of it, the auditory part of it will be stored separately in those relevant areas in our brain, in the olfactory cortex, in the auditory cortex, in the sensory areas and all. One memory will be split up and stored in all these areas, but our brain is able has already formed connections so that when one of these stimuli come in, it can pick up on all those memories and bring them together to form a to show us a complete memory and to react based on that. So the semantic memory is mainly a function of lateral and anterior temporal cortex and the prefrontal cortex, whereas the hippocampus, medial temporal lobe, and neocortex are what are responsible for the episodic memory. Okay, so moving on. Now we see it how we classify memory based on the permanency of storage. That is, there is short-term memory, intermediate, long-term, and long-term memory. So short-term memory, as I told you, something that lasts from seconds to minutes. And there are two possible mechanisms that have been given to explain how the short-term memory could be formed. One is signals travel around and around a temporary memory trace in a circuit of neurons. So what do we mean by the memory trace? That is this small association that you've created, that you looked at a phone number, you read the number, you might repeatedly read it to yourself, and you also that there is some kind of auditory component to it. Sometimes you would not be reading it aloud, but rather reading it in your mind only. And along with that, there's a visual component. You're trying to create a photographic or picture memory of it. So these are temporary memory traces that are formed in your brain. And the signals that again and again go around this loop keep it alive for a time period. And afterwards, when the requirement is over, this stops and that memory is forgotten. That is one mechanism of short-term memory. Second is, as I told already, based on it could be possibly due to presynaptic facilitation or inhibition, like we spoke of in habituation and sensitization. Those processes, the presynaptic facilitation inhibition, postsynaptic facilitation inhibition are the main neural mechanisms that are involved in memories, in the various kinds of memories. So these are the two possible mechanisms of short-term memory. Working memory is another name that is given to short-term memory. So it is a memory used in the course of an intellectual reasoning. It is terminated as the problem is solved. You collect a few information from things that you remember from various parts of the brain, put them, it is something like the RAM of your computer, the random access memory. Okay, so you know that whatever program is running on your computer right now, my Microsoft PowerPoint and Zoom is working on my RAM. So we increase RAM now so that I can multitask or run more programs without crashing or stuck up. Same way, those memories that are needed are brought to use. Whatever new input is brought together, and reasoning is done, solutions are found out, and usually after that, unless this is something that you need later on, unless your emotions perceive that this is something that I need later on, this is life altering, this is increase my skill, or this is life altering for the human race, unless some thoughts like that come up, these will be lost. They will not be retained after that. That is why it's called working memory also. So it's mostly processed in the prefrontal cortex. It's connected to hippocampus and parahippocampal regions also. And for this, directed attention is necessary. That is, you have to be focusing on it to get this done. If your attention is distracted and between to something else, you might easily forget that. Simple is when you're trying to count something together. You have a bundle of 100 notes in your hand. You're doing a sequential counting. And the moment your attention is distracted from that, you might lose your count. To prevent losing that attention, even when we're looking somewhere, we might be audibly counting also. We might count out aloud so that we don't miss the numbers. Some way can be done. So you are paying attention to that by counting the numbers, even though your eyes are looking elsewhere. If your attention is taken away from the working memory, it will be lost. And dopamine is one of the neurotransmitters involved in this. Now, intermediate long-term memory is that which lasts for minutes to weeks. And the process that is happening here is temporary chemical or physical changes, or both, in the presynaptic or postsynaptic membrane. That is, temporary or permanent changes can happen. These could be just chemical changes like increased calcium retention, increased release of neurotransmitters, or physical changes can happen. That is, the area of contact between the presynaptic and postsynaptic membrane may be increased. There may be more areas of for the release of neurotransmitters, the active zones. You would remember when you studied the synapse. So the area of the active zone would be increased. So chemical or physical changes can happen, and this could be either these will usually be temporary. That is, these changes will be reverted after weeks if that particular association that is created between the synapses is not being frequently used or reinforced. Okay, so that is intermediate or long-term memory from minutes to weeks, and temporary chemical or physical changes, or both, in the presynaptic or postsynaptic membranes. So two theories are used to explain the changes that happen in the presynaptic and postsynaptic membranes. So one is the presynaptic facilitation theory. That is, it was studied by Kandel and his colleagues. The idea was that there is a facilitator neuron. If you look at the diagram on the right side, you will see there's a presynaptic neuron that is coming here, and there is a this is the unlabeled one, is the postsynaptic neuron. And at the presynaptic neuron, a facilitator neuron is ending. This facilitator neuron will secrete serotonin. This serotonin will act on serotonin receptor, which activates adenyl cyclase, increase formation of cyclic AMP here. The cyclic AMP activates the protein kinase A, which will block potassium channels. We know that when a depolarization has happened, after that it is followed by repolarization. We have studied the various muscle and nerve repolarization in cardiac and skeletal muscle, etc. So all those graphs, when they went up, they come down afterwards. That is, when depolarization happened due to influx of calcium ions, etc., then there will be influx of potassium ions following that, which will cause the membrane potential to come back to the resting membrane potential. So this process, due to the involvement of serotonin here in the cyclic AMP system through protein kinase A, it stops the potassium conductance there. So the calcium ions continue to be built up inside the presynaptic membrane, and when there are more calcium ions in the presynaptic membrane, more neurotransmitter is going to be released for a longer time before the repolarization happens, and so the signal conductance is going to be for stronger and for longer time. So that is the presynaptic facilitation theory studied by Kandel and colleagues. So it is due to the facilitating neuron, a facilitating terminal in the presynaptic neuron, that causes longer and stronger or more release of neurotransmitter from the presynaptic to the postsynaptic neuron. Another theory was proposed by Byrne and his colleagues. They are saying that stimuli from separate sources acting on a single neuron under appropriate condition can cause long-term changes in the membrane properties of postsynaptic. So in this earlier one, that is in Kandel and colleagues, it was a single neuron acting on a single postsynaptic neuron. The presynaptic neuron was influenced to act stronger. Here Byrne and colleagues are talking about multiple neurons. So something like what we talk about the synaptic properties, we talk about convergence, something like that. So multiple neurons, when they are acting on a single postsynaptic neuron under suitable conditions, when they are firing together, etc., the changes will happen in the postsynaptic membrane. Some chemical or physical changes will happen in the postsynaptic membrane that will cause long-term memory. Earlier it was presynaptic changes. Byrne and colleagues suggested that it will be due to postsynaptic changes. So what are we talking about here? Short-term memory, we said these are things kept in loops. After some time, they'll be lost. Now we are talking about intermediate long-term. That is, that short-term memory now needs to be remembered for minutes to weeks. So some changes are being made in the synapses, so that that association that was formed, that is, when I smell that thing, it is something that I want to move away from, or when I smell that thing, that is delicious food, I feel like eating it. So some memories that are now need to be stored for longer, so that association between that olfactory stimulus and my appetite or my emotions is being created there. So it could be intermediate or long term. So this is the changes that usually happen in the pre or postsynaptic membranes. Now coming to long-term memory, as I told you, it is stored in the various parts of the neocortex. It involves not only chemical changes at synapse, but mainly structural changes. So earlier I told you about chemical changes that can happen. That is prolonged action potential, prolonged calcium calcium being present in the presynaptic membrane for longer, and so more of neurotransmitter release. Those are chemical changes. But certain physical or structural changes can also happen. That is, increase in the active zone, more contact area between the pre and postsynaptic neuron, so that more neurotransmitters are released and absorbed there. So these structural changes. So when you come to long-term memory, these structural changes are more seen than the chemical changes. Just a minute, please. Yeah, so long-term memory involves more of structural changes. And the most important of structural changes that we see here is, one is there is increase in the vesicle release sites from the presynaptic membrane. So more vesicle release sites for secretion of transmitter substance. Second is increase in the number of transmitter vesicles released. So there are more sites, more area for release, more number of vesicles are coming to these sites to be released. Then third is there are increased number of presynaptic terminals. That is, we are talking about one signal coming in from one neuron transmitted to one postsynaptic neuron. There will be growth of more neurons also, more synapses occurring from multiple synapses onto this new synapse through which the effect has to happen. So increase the number of presynaptic terminals can happen. Then changes in the dendritic spines that permit transmission of stronger signals. So various structural changes that happen in these spines that result in transmission of stronger signals. So these are 4 changes, physical structural changes that we see in long-term memory. So increase in vesicle release sites for secretion of transmitter substance, increase in the number of transmitter vesicle released, and increase in the number of presynaptic terminals, or changes in structures of the dendritic spine so that stronger stimulus can be transmitted. These are the various changes we see in the structural changes we see in long-term memory, apart from those we mentioned along with the intermediate long-term memory. So with that, we come to the conclusion of the various classification of memories we saw. We saw there was implicit and explicit. Then we saw there was short-term or working memory, short, intermediate, and then long-term memory. We saw the various molecular mechanisms that are active there, various changes that we see as acting there. Now, what do we mean by consolidation of memory? Consolidation of memory is the process that's essential for the formation of long-term memory. So short-term memory, if activated repeatedly, will initiate chemical, physical, and anatomical changes in the synapse that will result in the long-term memory. So a repeated thing, something that I just read that phone number once and made a phone call. Then I realized that then afterwards, after some time, I need to call that person again. So again, I look it up and make a phone call. It happens so that I have to continue doing this for 10 or 20 times that day. At the end of the day, high chances that I will remember that phone number by heart. The all 10 digits of it I'll remember. Same way, if it's associated with other things. For example, I'm trying to learn the new phone number of my wife. So this is something that I'll need always, require always. So instead of just putting that into a temporary memory into making a call, I will put more. There is an emotional component associated with the brain senses that this is something important for me. So instead of just putting it into short term, the associated memories together will make it into a long-term memory. So one of the ways that to do it is by repeatedly doing it. Like when we do studying, we repeatedly read something to study. Another is associated other sensory inputs. Like when you guys have all cleared your entrance and come, now you've been studying for your exams also. So what do you do? Some of you will read repeatedly. Some of you will write it down after reading it once. So these two inputs help solidify. Some of you like help to remember better by listening to somebody else teaching you. So you'll have group discussions and somebody teaching you something helps you understand the concepts better and learning. So all these are the different ways by which the memory is consolidated. Okay, so you around 5 to 10 minutes usually required for minimal consolidation to start happening. So repeatedly, if you're trying to do something, you'll require around 5 to 10 minutes to get that, commit that to memory. Or 1 hour or more spend on that topic or on the subject will help you to get a more strong consolidation of memory. So what is consolidation of memory is a question that might be asked for you, whether in Viva or and all will be used. You'll be asked, what is what do you mean by consolidation? So it's the conversion of short-term memory to long-term memory. If activated repeatedly, it causes various chemical and physical changes. And this is the function of the hippocampus that we studied in the case of that patient. So he was lacking in this, so he couldn't consolidate his memories. He could pick up a lot of new things. He could pick up skills even. Skills are not passed through the hippocampus. So it was through the striatum and all. So he could pick up even new skills, but he could not consolidate his memory because he didn't have a hippocampus and the medial temporal lobe was removed. Now, what are the features of consolidation? The new memories are codified into different classes of information. What do you mean by that? So when your short-term memory is being confirmed in the long-term memory, as I told before, it is classified according to the various sensory modalities involved, and depending on what kind of information. It could be something you're professional, it's something personal or family related. Various kinds of compartments our brain creates for all these things, and accordingly retrieval becomes easier and faster. That is why this proper classification and codification is happening. So during this process, similar types of information are pulled from the memory storage pins and used to help process the new information. When you're learning something new, for example, you're learning physiology. In physiology, you're learning something in CNS, you're learning memory now. So you will your brain will try to understand and store it if as you're taught, as you understand that this is a function of hippocampus, neocortex, etc. Your brain will try to associate it with the knowledge of anatomy of the cortex that you have, the knowledge of physiology of the cortex that you have, the various neurotransmitters, all these things that you have learned. Your brain will associate this with that, so that this helps in faster and proper retrieval of this memory later on. And rehearsal enhances the transfer of short-term memory to long-term memory. As I said, repeatedly going through that will help you to transfer the memory from short-term to long-term memory. These are the features of consolidation. Now, another thing that you need to know is the intercortical transfer of memory. It simply means, as the term indicates, the transfer of memory from one cortical hemisphere to the other. For example, if you are looking at a diagram and your one eye is closed all the time while you're looking at it, you looked at it, you drew it and everything. After you finished drawing or after you finished learning that, if you close the other eye, and only the let's say initially the left eye was closed. Now, if you close the right eye, if you look at the diagram, you still know what that diagram was. You will still remember, let's say it was a diagram of the pyramidal tract. You will still recognize the various structures there, the medulla, the pyramids and everything. You will recognize that. You will not have any problem in understanding or recognizing that. Arthana, could you please switch off your mic? Thus, memory is transferred from one cortical hemisphere to other. So this was mainly understood by experiments from cats and monkeys and all. One eye of the animal was blindfolded and they were exposed to something, and later it was seen that even if you close this eye with the other eye also, the cat is able to understand, perceive, and react according to the memory that was created already. Then Artana, please switch off, mute your mic. Yeah. So this intercortical transfer happens through the connections between the two cerebral hemispheres, that is the corpus callosum and the anterior and posterior commissural fibers. These are the structures through which the intercortical transfer happens. Mostly this is a function of the corpus callosum, and it has been experimentally found that creating lesions or cutting off these connections will result in memory not being transferred from one side to the other. That is, in the animal, if you're exposed to with one blindfolded eye and later on you take off the blindfold, the cat will not be able to understand that the other eye, and it is known as a split brain animal in these experiments. That is, when you split the two hemispheres into two, there will be no more transference of information from one to the other, and so what was perceived with one side is not being able to be perceived by the other side. That is what is known as a split brain animal. So this is intercortical transfer of memory. Another question that can be asked for you. So just to sum it up all, explicit memory, it is mainly a function of medial temporal lobes, short term in the hippocampus. Short term working memory involves both the hippocampus and the prefrontal. If it has to go, when it becomes long term memory, it is stored in the neocortex. The hippocampus is involved in this process. Without the hippocampus, the conversion from short to long term will not happen. Then implicit memory, priming is a function of neocortex, procedural mainly striatum, associative involves amygdala and cerebellum, non-associative involves the reflex pathways. Then synapse is the physical site of most memory. And then biochemical basis, new protein formations, that is, we told about how long-term memory is formed. There has to be physical structural changes happening. So new protein formation is involved, increased mRNA formation, and the most important transmitter involved in memory is acetylcholine. Most of these structures we talked about, the hippocampus, the prefrontal cortex, and various areas, the connections are basically through cholinergic neurons. And because of this, certain diseases that affect these neurons more will cause loss of memory. So these applied aspects, I want you to read up by yourself. These are again questions that can be asked as short notes for you also. So based on what you have learned, this is all there in your textbook. Just read it up and you prepare short notes for yourself. This is a assignment for you. Okay, so you need to look up amnesia. What is Alzheimer's disease? Look at what is happening in Alzheimer's disease. What are the treatments available? What are the drugs that are facilitating memory? What is confabulation? What is strangeness and familiarity? The process involved in what we know as deja vu. So these are the applied aspects of it. I am not going to teach that. I want you to read it up from your textbooks. There is amnesia, Alzheimer's disease, drugs facilitating memory, confabulation, strangeness, and familiarity. So with that, we come to the end of this class. Your classes will be continued by Smriti ma'am now. Please, please do not leave the classroom. Okay, thank you, class. Please do not leave. Smriti ma'am will be joining now.
Thank you, sir.
Thank you, sir.
Good morning. Am I audible?
Good morning, ma'am. You are audible.
Okay. So good morning, everyone. Next topic for the class is reticular formation. So shall I start the class?
Can we have a 5-minute break?
Okay, you need 5 minutes break. Okay, then we'll start by 11:10. Now it's 11:05. We'll start by 11:10. Okay. Okay, we'll start the class. I hope everyone is back. So the topic for this session is reticular formation. Now, what do you mean by reticular formation? The word reticulum, it denotes a network of neurons or nerve fibers that are closely located within your brainstem. Now, in your previous classes, you might have come across different named pathways, isn't it? You might have learned about the sensory pathways, the ascending pathways like the spinothalamic tract, then the motor pathways like your pyramidal tract, which is a descending tract, and so on. So reticular formation mainly involves those neurons and nerve fibers that are left behind after all these nerve named fibers and neurons have been accounted for. So coming to the objectives of this class, you need to know about the different neuronal aggregates involved in reticular formation, the different connections and functional divisions of reticular formation, then the important functions of reticular formation, and finally, we'll see few points related to the applied physiology. So, coming to the introduction, as I have told you, reticular formation is a complex network of neurons and nerve fibers that are scattered throughout the central part of the brainstem. It is mainly formed by neurons and processes that are left over after all the well-defined nuclei and pathways have been accounted for. So structurally, the brainstem reticular formation mainly consists of neuronal aggregates, afferent connections, and efferent connections. So we'll see each one of them. What are the neuronal aggregates involved in reticular formation? So neuronal aggregate means it includes a number of reticular nuclei that can be divided into 3 longitudinal columns in each half of the brainstem. So the 3 different longitudinal columns are, you have got nuclei of median column, nuclei of medial column, and the nuclei of lateral column. So these are the different neuronal aggregates. So we'll see which are the nuclei involved in each of these longitudinal columns. Coming to the nuclei of median column. Now this nuclei of median column lies close to the midline. So imagine the brainstem, there is a midline, and just close to the midline lies the nuclei of median column. It is called as nuclei of Raphe. And one example of a nuclei present in this column is Raphe nuclei in the midbrain. So that is regarding the nuclei of median column. Now coming to the nuclei of medial column. Now nuclei of medial column lies just lateral to the nuclei of median column. And this column is mainly made up of large cells or magnocellular cells. And examples are nucleus gigantomedullaris in the medulla and the pontine tegmental nuclei. These are the examples of nuclei which you can find in the medial column. So we have seen nuclei of median column and the nuclei of medial column. Now coming to the nuclei of lateral column. Now nuclei of lateral column lies just lateral to the medial column. And this lateral column is mainly made up of small neurons or parvocellular neurons. And examples are the central nucleus of medulla and the central nucleus of pons. So these are the different types of neuronal aggregates. So you can divide them into 3 longitudinal columns. So in the brainstem, just close to the midline, you have got the nuclei of median column. Lateral to that, you have got nuclei of medial column. And lateral to the nuclei of medial column, you have got nuclei of lateral column. So these 3 columns forms the 3 longitudinal columns that are seen in each half of the brainstem. Together, they form the neuronal aggregates that form a part of the reticular formation. So this was the structural classification of the neuronal aggregates. Now functionally also you can have neuronal aggregates, and these include the cardiac centers, respiratory centers, vasomotor centers, and the chemoreceptor neurons. This is the functional classification of neuronal aggregates. So that is in short regarding the neuronal aggregates involved in reticular formation. Now we'll move on to the connections of reticular formation. So you have got afferent connections and efferent connections. Now, efferent connections are further divided into two systems. You have got an ascending reticular activating system and a descending reticular activating system. So first of all, we'll see the afferent connections. What do you mean by afferent connections? What is the meaning of afferent?
Those connections that reach the reticular formation from various parts of the body.
Yes. So here, afferent connections can mainly come from the spinal cord through the spinothalamic tract. Can you give an example of spinothalamic tract? You have got anterior spinothalamic tract and lateral spinothalamic tract, isn't it? So afferent connections can pass via spinothalamic tracts, like your pain sensations, which are carried by the type C fibers. That is one example for afferent connection to the reticular formation. Also, afferent connections can come via collaterals from all the ascending tracts, like the trigeminal pathway carrying sensations from the face, then your visual pathway, auditory pathway, gustatory pathway, vestibular pathway, etc. So all these pathways are ascending tracts that can give afferent connections to the reticular formation. Again, you can have afferent connections from the brainstem afferents from their cranial nerves. Also, afferents can come from the entire cortical areas and subcortical structures like basal ganglia, hypothalamus, thalamus, and cerebellum. So these are some of the important afferent connections of the reticular formation. So here you can see this is a schematic representation of the various afferent connections. You can see that afferents are coming from various sensory pathways from the spinal cord through the pathways for touch, pain, temperature, and kinesthetic sensations. Then you can see afferents coming from thalamus, corpus striatum of the basal ganglia, cortex, as well as from the cerebellum. So these are the different afferent connections. Now coming to the efferent connections. So efferent connections, again, it can go to the spinal cord as the descending reticulospinal tract. Again, efferent connections can also go to all the nearby structures from which afferent fibers are coming to the reticular formation. Also, a major long efferent fibers can go to the cerebral cortex. So again, in this diagram, you can see efferent connections going to the cortex, cerebellum, to thalamus, hypothalamus, then to the basal ganglia, especially to the red nucleus, tectum, and substantia nigra, and to the spinal cord through the reticulospinal tract. So these are the different areas to which efferent connections are going from the reticular formation. So from this, you can understand that reticular formation means it is an area involving highly interconnected neurons and nerve fibers. There are both afferent as well as efferent connections involved within the reticular formation. Next, we'll see how the reticular formation is divided functionally. Function-wise, you have got two systems. There is an ascending reticular activating system, which is commonly known as the ARAS, and then you have got a descending reticular system. The descending reticular system is further subdivided into a descending facilitatory reticular system and descending inhibitory reticular system. So these are the functional divisions of the reticular formation. Now, among these systems, the ascending reticular activating system is very important from your exam point of view. So you may get a short note to write down the functions of ascending reticular activating system. This system is again important in the physiology of sleep and wakefulness. So we'll see each system. Coming to the ascending reticular activating system. So this system will receive collaterals from specific ascending pathways. So to understand the ascending and descending pathways, you need to know the different ascending and descending pathways of the brain. So ascending reticular activating system, it will mainly receive collaterals from the specific ascending pathways. One example is the spinothalamic tract. Also, this ARAS can receive collaterals that will funnel from the trigeminal pathway, visual pathway, auditory pathway, and olfactory pathway. So all these are ascending pathways that can form a part of the ascending reticular activating system. Now, most of the ascending fibers to the RAS will mainly rely on non-specific nuclei of thalamus. Can you name the non-specific thalamic nuclei? Class on thalamus is already over, isn't it? Which are the non-specific thalamic nuclei? You have got intralaminar and midline nuclei. So most of the fibers that are ascending to the RAS will rely on this non-specific thalamic nuclei, and then they will diffuse to different parts of the cerebral cortex. So here you can see in this diagram, you can see the area marked as ascending reticular formation. And here you can see the fibers, various fibers ascending up from the spinal cord through the different afferent pathways. Some of the fibers are relying on the thalamus. Other fibers are diffusely spread to different parts of the cerebral cortex. Now, what is the basic difference between this ascending reticular activating system and the classical sensory pathways? See, you can see from the diagram itself that the RAS or the ascending reticular activating system can be stimulated by number of impulses. Any impulse that is arising from any of the ascending sensory pathways can stimulate the ARAS. So it can be excited by any type of sensation. So you can call this ascending reticular activating system as a non-specific system. Also, the ARAS fibers, that is the ascending reticular activating system fibers, occupy the core portion of the brainstem. On the other hand, if you take the case of classical sensory fibers, the sensory pathways, these pathways are highly specific and they can be activated only by one type of sensory stimulation. For example, your lateral spinothalamic tract, it can be activated only by the pain and temperature sensations. Similarly, visual pathway can be activated only by the visual sensations and so on. And another important aspect is these classical sensory fibers are mainly occupying the lateral aspects of the brainstem, whereas ascending reticular activating system is occupying the central core of the brainstem. So ARAS is mainly concerned with non-specific sensations, whereas the classical sensory pathways are concerned with specific sensory sensations. So now we'll see what are the functions of the ascending reticular activating system. So the first important function is to maintain a state of wakefulness, alertness, and consciousness of an individual. This is mainly by sending strong stimulatory fibers to the major neurons present in the brainstem. So what this ARAS does is it will send strong facilitatory drive to the central neurons in the cortex, which can raise the background excitability and increase their responsiveness to specific stimuli. So ascending reticular activating system is mainly concerned with maintaining a state of wakefulness, alertness, and consciousness of an individual. So whenever this RAS is stimulated, there is wakefulness and alertness of an individual, which is required for proper sensory perception. So if you want to perceive any type of sensation, you need to be awake. And for an individual to be awake and alert, the RAS, that is the reticular activating system, has to be in a stimulated state. Conversely, when the impulses coming from the reticular activating system are decreased, there is neuronal fatigue and the person will go into sleep. So always in the wakefulness state, the RAS will be activated. Whenever the impulses through RAS is declining, the person will go into a state of sleep. So that is the first important function, maintenance of state of wakefulness, alertness, and consciousness of an individual. So if you're alert and active in my class, it indicates that your RAS, reticular activating system, is stimulated. Now moving on to the next function, RAS or the reticular activating system also helps to maintain the cortical neuronal excitability. So how can you assess this? How can you assess the electrical activity of the cortex? Is there any recording that you can take to assess the electrical activity of the cortex?
EEG.
Yes, very good. You can record an EEG, and this will tell you the EEG pattern will tell you the amount of firing that is occurring through the reticular activating system. So that is the second function, maintenance of the cortical neuronal excitability. So fibers from the RAS will synapse with the dendrites of the cortical neurons, thereby increasing the excitability of the cortical neurons. Now, the thalamic reticular neurons are also capable of this intrinsic spontaneous activity. So we can say that RAS is closely related to the electrical activity of the cortex, and this can be recorded from EEG. And an EEG pattern that is taken at rest will give you an idea about the rate of firing through the reticular activating system. So suppose if the reticular activating system is stimulated, which wave or which rhythm can you find in EEG? You might have learned about alpha, beta, delta, and the theta waves, isn't it? So stimulation of the reticular activating system is marked by which rhythm in EEG?
Beta.
Beta.
Yes, very good. Beta rhythm of EEG. So that is another importance of this reticular activating system. Now, moving on to the third function, reticular activating system also plays an important role in appreciation of sensations. This we have already seen while discussing the different connections. You know that most of the ascending pathways, they will give collaterals to the reticular activating system. So a person can perceive a particular sensation only if the reticular activating system is active. One example is the pain sensation. So during sleep, all the sensations will be dull because during sleep, the reticular activating system will be in an inhibited state. Next is the role in selective attention and sensory inattention. So you might be familiar with these terms from your practical classes. So thalamic nuclei of reticular formation is responsible for this ability to concentrate or focus on a particular sensation. So the field of awareness is mainly narrowed by facilitating certain cortical areas and simultaneously suppressing certain other areas. So a classical example for this is the soldiers in the battlefield, they do not feel the pain until the battle is over. This is mainly due to the property of selective attention and sensory inattention. So that is the fourth function. So these are the different functions of the ascending reticular activating system. So whenever you hear the term RAS, it mainly denotes the ascending reticular activating system. Okay. So we have seen the important functions of RAS. They include the maintenance of a state of wakefulness, alertness, and consciousness of an individual. Then RAS is important for maintaining the cortical neuronal excitability. It is important for appreciating the sensation, and also it plays an important role in selective attention and sensory inattention. That is, selectively focusing on particular sensations and avoiding other sensations. Okay, so these are the exclusive functions of the ascending reticular activating system. Now we'll move on to the descending reticular activating system. So this mainly includes the descending fibers from the reticular formation, which mainly forms the reticulospinal tract. So you might be familiar with this reticulospinal tract from your class on motor system. You might have come across this tract while learning about the physiology of muscle tone. So this system mainly includes two kinds of reticular projections. You have got a descending inhibitory reticular projection and a descending facilitatory reticular projection. So we'll see the location and function of each. Coming to the descending inhibitory reticular projection. Now the inhibitory reticular projection, it is mainly located in the ventromedial part of medulla, and fibers from this area mainly descend to the spinal cord and end in the interneurons, thereby inhibiting the motor neurons. They receive an inhibitory input from the descending fibers of the cerebral cortex and basal ganglia also. So what is the function of this descending inhibitory reticular projection? It causes reduction of the movements because it is an inhibitory projection. So it will cause reduction of the movements either reflexly by pyramidal stimulation or by stimulation of the motor cortex. So remember, descending inhibitory reticular projection is located in the medulla and its function is to inhibit the motor neurons. In addition, inhibitory input is also coming from the cerebral cortex and basal ganglia, and the function is to cause reduction of movements. Now coming to the descending facilitatory reticular projection. Now the facilitatory reticular projection, mainly the fibers of this area will send facilitatory fibers to the spinal cord neurons. Now these fibers are mainly located in the pons. So you also call this area as bulboreticular facilitatory area. And impulses coming from these areas will mainly influence the gamma motor neurons. Now, since they are facilitatory fibers, they will cause facilitation of the cortically or reflexly induced movements. Another nuclei that can produce facilitation is the vestibular nuclei via the vestibulospinal tract. So to summarize the descending reticular activating system, you have got mainly two projections, a descending inhibitory reticular projections and a descending facilitatory reticular projections. The inhibitory reticular projections mainly located in the medulla. They will mainly inhibit the motor neurons and cause a reduction in the movements. Also, inhibitory inputs are arriving from the cerebral cortex and basal ganglia, also from the cerebellum. Again, descending facilitatory reticular projection is located in the pons. So it is also called as the bulboreticular facilitatory area. And this area, the fibers will mainly influence the gamma motor neurons and cause facilitation of movements. Vestibular nuclei is another neuron that is influencing the spinal cord neurons and causing facilitation through the vestibulospinal tract. So you might have come across this facilitatory and inhibitory reticular projections while learning about stretch reflex. So I'll show you a diagram to summarize the facilitatory and inhibitory inputs. So here you can see this area is the excitatory reticular projection area located in the pons, and this is the inhibitory reticular formation area located in the medulla. So in the excitatory area, you can see that excitatory impulses are also coming from the vestibular nuclei, whereas inhibitory influences also arise from the motor cortex, basal ganglia, and cerebellum. So facilitation is mainly in the pons. Facilitation is mainly from the excitatory reticular formation along with the vestibular nuclei, while inhibition is located in the medulla, along with the inhibitory influences from the motor cortex, basal ganglia, and cerebellum. So together, these two will form the reticulospinal tract, and they play a very important role in regulating your muscle tone. The balanced activity from both these excitatory and inhibitory projections will help you to control the muscle tone. So now we will see what are the exclusive functions of this descending reticular activating system or the reticulospinal tract. So as I have told you, one important function is the control of muscle tone. Now gamma motor neurons, they have connections from both the facilitatory and inhibitory reticular projections, as you have seen in the previous figure. So a balanced activity of both these projections will help in the control of smooth and purposeful movements, thereby controlling the muscle tone. So that is one important function of the reticulospinal tract or the descending reticular activating system. Another important function is the maintenance of posture. So the reticulospinal tract, it will facilitate the spinal stretch reflexes by exciting the extensor group of muscles, which in turn will help in the maintenance of posture. Also, the descending reticular fibers can regulate the different postural reflexes. So the two important functions of the descending reticular activating system are maintenance of tone and maintenance of posture. So that concludes the functional divisions of the reticular formation. So under functional divisions, we have seen that there is an ascending reticular activating system and a descending reticular activating system. Under ascending reticular activating system, the important functions are to maintain a state of alertness, wakefulness, and consciousness of an individual, to maintain the cortical neuronal excitability. Then it has got a role in the perception of different sensations and a role in sensory attention, selective attention, and sensory inattention. Coming to the descending reticular activating system, it mainly includes the reticulospinal tract. It has got two types of projections, a facilitatory projection and an inhibitory projection. And the main functions are to maintain the tone and posture. So we have seen the exclusive functions of ascending reticular activating system and the descending reticular activating system. Now to summarize, we'll see the overall functions of the reticular formation. So as you have seen, it has got a role in sleep and wakefulness, in selective attention and sensory inattention. It regulates the transmission of various sensory stimuli. It plays an important role in the control of muscle tone as well as in the regulation of postural reflex changes. Then some extra functions are there, which is common to both systems. One is the conditioning and learning mechanisms. So this reticular formation, it can act as a neuronal substrate for various conditioning reflexes as well as for the learning mechanisms. Again, another additional function is the control of visceral functions. Now, reticular formation will influence the visceral functions mainly via its hypothalamic connections. So the important visceral functions that are controlled by reticular formation are the vomiting and swallowing reflexes. Both these reflexes are mainly integrated in the medullary reticular formation. So if you remember your class on vomiting and swallowing reflexes, you can recollect that the major integration is occurring within the medullary reticular formation. So it has got a role in controlling the visceral functions. And finally, it plays an important role in the cardiovascular and respiratory functions. You have already seen under respiratory and cardiovascular physiology that important cardiovascular and respiratory centers are located within the brainstem reticular formation. Now, cardiovascular centers like the vasomotor center, it is located within the medullary reticular formation, and it plays an important role in the regulation of heart rate, cardiac output, vascular tone, and thereby plays an important role in the regulation of blood pressure. So cardiovascular centers are also integrated with the brainstem reticular formation. Again, the respiratory centers like your inspiratory and expiratory group of neurons are also located within the central gigantocellular cells in the medulla. So they are also regulated within the medullary reticular formation. And the pneumotaxic and apneustic centers are mainly located within the pontine reticular formation. So the cardiovascular and respiratory functions are also influenced by the reticular formation. So these are the different functions of the reticular formation. Apart from its role in controlling the sleep wakefulness cycle and sensory perception and control of muscle tone and posture, it also plays an important role in controlling the visceral functions, cardiovascular and respiratory functions, and also it acts as a neuronal substrate for the conditioning reflexes and learning mechanisms. Now, there are certain neurotransmitters involved within the reticular formation. So you have got the cholinergic neurons, large cholinergic neurons, which will project to the cortex via the thalamus. Then you have got adrenergic neurons that project via the intralaminar nuclei of thalamus. Then there are noradrenergic neurons projecting to the cerebellum and dopaminergic neurons projecting to the thalamus, cerebral cortex, hypothalamus, and limbic structures. So these are the different neurotransmitters that are related to the reticular formation. Cholinergic neurons, adrenergic and noradrenergic neurons, and the dopaminergic neurons. Now coming to the applied physiology. You have got certain drugs or chemical conditions that can stimulate the reticular formation and some drugs that can inhibit the reticular formation. So drugs which excites the reticular formation, they will mainly maintain the person in an alert and awakeful state. So alertness and arousal is mainly brought about by sympathomimetic drugs like adrenaline and noradrenaline. Then acetylcholine, it can specifically increase the cortical activity and thereby excite the reticular formation. Then chemical conditions like increase in carbon dioxide or an increase in blood pressure can also cause excitability of the reticular activating system. And drugs which inhibits the reticular formation means they are decreasing the alertness level, or they are putting the person into a sedative or sleeping stage. So examples will be general anesthetics and sedatives. So this forms the basis of using anesthetics prior to any surgeries. They can put the person into a inhibitory state by inhibiting the reticular activating system. So to summarize, we have initially seen the different neuronal aggregates involved in reticular formation, like the nuclei of median column, medial column, and the lateral column. Then we have seen the different afferent and efferent connections related to the reticular formation. Then we have seen what is ascending reticular activating system and its functions, and the descending reticular activating system and its functions. Then we have seen what are the other functions related to the reticular formation, like the control of visceral functions, respiratory and cardiovascular functions, and role in learning and conditioning. Then we have seen the important neurotransmitters of reticular formation and the drugs that can excite and inhibit the reticular formation. So the questions that you can get frequently from this part is either you can get a short note on reticular formation, wherein you will have to mention the functions of ascending and descending reticular tracts as descending reticular activating systems along with the other common functions, or you may be asked about ascending reticular activating system alone. It can be asked as a separate short note. So any doubts? Okay, then we'll wind up the class. Thank you.
Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.